Welcome to this explainer. Okay, let's dive into this. We hear the terms DNA and RNA thrown around constantly, right? Whether it's in the news, biotech, or healthcare, they are everywhere. But today, we're going way beyond just the basic acronyms. We are going to look at the absolute foundation of modern genetics. We're talking about the ultimate biological partnership, the blueprint and the builder.
So think about this for a second. What actually builds the human body? It's kind of a profound question, and to answer it, we really have to look past the simple idea of genetic destiny. You know, we constantly hear that our genetic code dictates exactly who we are. But honestly, a static code just floating around inside a microscopic lipid bubble doesn't equal life. Think about how a real-world construction site works. Having a pristine, perfect architectural blueprint is absolutely useless if there's no one there to actually pour the concrete and frame the walls. To turn inert biological information into a living, breathing, functioning organism, you need an active builder. You need a highly organized system that can read those instructions, interpret them, and literally execute them. That right there is the fundamental problem biological life had to solve just to exist in the first place.
Here is our roadmap for today. First, we'll look at the molecules of life. Then, DNA, the master blueprint, followed by RNA, the working copy. After that, we'll compare the blueprint versus the builder, and finally, wrap up with why it all matters today.
All right, starting off with Section 1, the molecules of life.
So, DNA and RNA. These are two vital molecules that carry genetic information, but they perform entirely different roles. And what's really fascinating here is thinking about how they actually evolved to share this massive genetic workload. You see, early life on Earth likely started in what scientists call an RNA world. Basically, back then, one highly versatile molecule tried to do absolutely everything, both storing the genetic code and executing all the vital chemical reactions. It was doing the whole shebang. But over billions of years, life evolved a much more sophisticated division of labor. By separating the long-term data storage from the daily chaotic action of the cell, biological systems achieved this incredible level of molecular stability. And that stability is exactly what eventually allowed complex, multicellular organisms like us to thrive.
Moving right along to Section 2, DNA, the master blueprint.
DNA is literally the ultimate long-term storage medium. It's the permanent blueprint containing the essential instructions for growth, development, and survival. But let's look at why it is so perfectly suited for this job at a chemical level. The D in DNA stands for deoxyribose, and what that means is its sugar backbone is literally missing an oxygen atom compared to RNA. Now, in organic chemistry, oxygen is highly reactive. So by removing that single oxygen atom, DNA becomes incredibly stable and non-reactive. Plus, it naturally forms that famous double helix shape, right? It tucks its vital, delicate information bases safely on the inside, completely shielded away from the messy, chaotic chemical environment of the rest of the cell. It acts just like an uncorruptible, permanent hard drive locked safely in a vault, ensuring those original instructions are preserved perfectly for an entire lifetime.
Which brings us to Section 3, RNA, the working copy. So if DNA is the vault, RNA is the active, dynamic working copy. It is generated directly from the DNA to help cells actually produce essential proteins. But here is a brilliant and honestly pretty surprising biological insight. RNA's most important evolutionary feature is actually its extreme instability. Yeah, you heard that right. It's a temporary, easily destructible molecule completely by design. Because it holds on to that reactive oxygen atom we just talked about, it's highly vulnerable to rapidly breaking down. And that is an absolute cellular superpower. Think about it. If RNA were as stable as DNA, a cell would just continuously read a single instruction and endlessly overproduce proteins until it literally choked itself. RNA's structural instability means it delivers the message, builds the necessary protein, and then boom, it quickly degrades. That gives the cell precise, minute-by-minute control over everything it's doing.
Section 4, blueprint versus the builder. Now, what's really interesting about this slide is how we can put these two molecules head-to-head.
We have DNA storing genetic info, remaining highly stable inside the nucleus, versus RNA carrying those instructions, being intentionally less stable and helping to actually make proteins. When you analyze their traits side by side like this, it perfectly illustrates how physical form dictates biological function. DNA's rigid, double-stranded structure firmly anchors it inside the nucleus. It functions purely as a vast repository of data. RNA, on the other hand, being a flexible, single-stranded molecule, is agile enough to slip right through the microscopic pores of the nucleus and navigate the crowded cellular cytoplasm. Think of DNA as this massive, irreplaceable library reference book that absolutely cannot be checked out. RNA? It's the quick photocopy you stuff in your pocket to take down to the actual workshop.
Let's move to and see how this builds into what scientists refer to as the central dogma of molecular biology. Step 1, DNA. Step 2, RNA. Step 3, protein. This brilliantly illustrates the beautiful thermodynamic efficiency of life's workflow. It starts with transcription, where specialized enzymes temporarily unzip a tiny section of the DNA blueprint just long enough to transcribe its code into a fresh strand of RNA. Then comes translation. That's where the RNA travels to a ribosome, which is basically the molecular manufacturing plant of the cell, which reads the sequence and physically strings together amino acids to build a functional protein. This elegant flow of information, from permanent storage to disposable messenger to an active molecular machine, is literally the foundational mechanism powering every single living thing on Earth.
Okay, Section 5. Why it matters today.
RNA in modern biology plays a pivotal role in gene expression, disease research, vaccine development, and broad biotechnology. The profound implications of this explainer manifest right here in our most advanced medical breakthroughs. Take a look at the revolutionary development of mRNA vaccines. For decades, scientists really struggled to safely deploy certain therapies. But by fully leveraging our understanding of RNA's role as a temporary builder, the whole game changed. We essentially learned how to hand the cellular machinery a short-lived blueprint. The synthetic RNA instructs the cell to build a harmless viral marker, training our immune system perfectly. And then, because of that vital chemical instability we explored earlier, the RNA completely degrades and vanishes. It delivers incredible, rapid immunity without ever having to touch or interact with your permanent DNA vault. It's a true game changer.
Now, bioinformatics tools analyze both DNA and RNA data to decode genes, biological processes, and exact disease mechanisms. So the crucial point is that to truly harness this system today, we need massive, unprecedented computational power. I mean, the human genome contains over 3 billion base pairs of DNA. That is a mind-boggling amount of information. You simply cannot parse that astronomical level of biological data just by looking through a microscope. Today's researchers rely on advanced bioinformatics to process incredibly vast genomic data sets simultaneously. By mathematically mapping both the stable DNA blueprint and the real-time dynamic activity of the RNA builders, scientists can pinpoint exactly where and how a genetic disease originates. Computing power has finally given us the ability to read the cellular worksite in high definition. This leaves us with a massive question. With bioinformatics and RNA technology, how will editing both the blueprint and the builder shape the future of human health? As we've seen, DNA safely stores the master instructions of life, and RNA dynamically carries them out into the real world. Together, they are the undisputed foundation of biology. But listen, we have decisively crossed a threshold where we are no longer just passive observers of this process. With technologies like CRISPR allowing us to target and permanently correct the DNA blueprint, and advanced RNA therapeutics allowing us to deploy temporary builders on demand, the medical frontiers are wide open. I want to leave you pondering that staggering potential. Thank you so much for joining me for this explainer. Keep wondering, keep learning, and I'll see you next time.